So we will again do some of the coding, and it is going to take a while for the coding to load and some of the packages to install. So it is okay if you are not able to follow up, and you can get the recording and then do some more work with some more coding in referring to the recording. But before that, I need to know how many of you, yeah, exactly. Thank you, NG. And how many of you have good expertise or decent expertise in R and Python? Okay, beginner, excellent. So if you are comfortable, if you're a newbie, or if you know only to open up R or Python's environment, and then you want use a couple of lines of codes to maybe quickly plot a plot, or maybe just load a very small file, and yeah, basic knowledge, that's perfect. Okay. Okay, that is what we want. So I don't want to start from absolute basic, like what is a variable, what is a data frame. So everybody knows what a data frame is, and everybody knows what an array or a matrix is. So that is what we want to understand. So, okay, we are going to use the environment that we are happy to explore with Google Collaboratory or Google Colab Python notebook or notebooks. So this is a shareable link environment, which will run in Python and also in R's programming tool, or R's programming tool. So you can access this, you can have your own copy of this when if you have your Google link or Google account set up. So if you have not set it up, quickly you can do this by typing collab. Yeah, wait a second. I hope shares the code on the chat. Yeah, we will share the codes that we are going to explore today. And yeah, so wait a second. Let me give the link where if you type collab.to, and then you can go to Collab's environment. And for example, this is what you will see. And this will run both Python and R's environment, even though it is the extension shows that it is interactive Python notebook. So right now it is simply a Python notebook, and you can actually open up a new notebook from here, because you cannot save what they name it as or what you get as a scratchpad. So scratchpad, you cannot save the codes that is present in this scratchpad, or you can save a copy later. So open a new environment so that we enable the automatic saving tool. So everybody start working along with me. So we need to do this before even starting to get an idea of what we are going to discuss today, because some of the packages that we need to use will take long time to install, so that we can manage our time effectively by running through these installations before even starting to use this notebook. So are you here? If everybody here, everybody can see untitled 0.ipynb, and this is a Python notebook. And how do I know? Because I opened it. And if you are unsure of whether you are using a collaboratory notebook in Python or in R's environment, you can go to runtime, the menu runtime, and then you can come all the way to change runtime type. And then if you do not see any option just below the notebook settings or just above the hardware accelerator to change to Python or R, then this is a Python's notebook. So you cannot run R code in this. This is a specific Python's notebook. So this is fine. We don't need to install anything in Python's notebook. But if you, instead of using this, if you use the another link like collab.to-R, right? It can be lowercase or uppercase. It does not matter. If you do this and enter, and you can do this collab.to-R. And we are going to look at an exactly identical environment, right? And we will explore what to do and how to run, and what are the couple of settings that you need to make sure that it is presentable, and etc. So how are we going to run this while we are exploring some of the codes? So in this environment, this I started it by typing collab.to/R. So I know that it is an R's environment, but if you're unsure of landing up in someone else's collab and etc., you can find out by opening runtime again, and then using the option change runtime type. So in this change runtime type, you have an option that tells this runtime type can be changed from R to Python, right? So if you have that option, and if you have that option R enabled, then it is a Colab notebook under R's environment. So that's you will know this. Okay, so we cancel a save. It does not matter. We get out of this. So what is the power of using such a Colab's environment? I mean, one of the important advantages, for example, you can see at some of the copy of Colab notebook. So you can write some codes and get an output right below that. And such level of linking the output to a specific code is not very easy in any other development environment or tool that you are going to use. Even in RStudio, which I have been using, or which many people will be using for R's environment, you will have something like this, where you have a place where you can type the codes, and where you have a place where you can execute the codes and interact with the R console, and then where you have a separate section where your plots is going to be displayed. I have not loaded any plots, so it is not loading up anything, but this is where your plots are going to display. And at any specific time, you can view only one of the plots, and you have to go back and forth to check previous plot and the next plot. So there are specific advantages in using Colab-like setup, and people are actually using Colab-like setup in local computer also, like Jupyter Notebook and etc. And that will help you in analysis like this. You can compare and contrast the output, right one below the other, and check what is a specific code that makes this type of plot, so that you can come back to the code and relate. And another big advantage is the shareability of such notebooks or such setup. So if you click on share, and you can share this notebook with many people. You can see that this is a very popular notebook, and this has been shared already with many people of my collaborators and co-workers. Not in this case, because I opened it. This is a copy of this, so you did not see it. So you can add people and add groups here and share it with others, so that you can check between the versions and between the execution of the code along with your friends and collaborators and your supervisors and co-workers and etc. And you don't have to add everybody here. You can just make it make the access general access as anybody can access with the link, and copy the link and then share it with people like you, and then so that anybody who opens this notebook can get access to the codes and the outputs. So this shareability is very, very important advantage of Google Colab. So enough of Colab's advantage. So let's go back again to your R Colab. So everybody has opened your R Colab here. So if everybody has opened up your R Colab, I want you to run these and then forget about that Colab. We will come back to this Colab when it is until the time that we are here, then it would have completed our installation. So how would you run this? This is my R Colab, and then you copy it, and you open up a code block from here, right? If you see something like this, and then you open up a new code block by clicking insert a code cell below. So you press, and then inside this, you can just type or paste. So this is what I want everybody to type or paste inside their R's notebook. And if you do that, and then start this install command, or start this execute this code option, then you will see it see some output and see it installing all the packages one by one. So let everybody start. So I'm not going to do it one more time because I have already done it. And let me know is your installation running smoothly. So you just have to start it and it will install. If you get an error, right, like this, for example, if you get an error like Let it run. It will actually initialize before running. So connect and initialize before running. So if you get an error like invalid syntax or install option not found, then that would mean you are actually running this code. This is an R code. You are running this R code in Python's environment. So make sure that you are running it under R's environment. Is everybody there? Can I see some responses?
Sir, how to run this? I have inserted
You
on
have
the.
pasted it and press this triangle.
Okay, okay, sir.
It will start running. So you'll see it working out and you'll see some output, some option output here. Something like this, installing package and etc. etc. Okay. Okay, we will let it install. It's going to take a while to install Suret and UMAP and CLS environment, which we will use extensively. And yeah, while it is running, you will see that the code takes a red or a gray color code, and it will show you that it is ready to be used in this kind of color code, yellow or yellowish orange color code. So let's clear.
It has started downloading.
Okay, perfect. So everybody, it is working. So that is the case. Let me clean up some of these things. So we let this run, and then we will come back to this later. Leave, that's okay. And I also don't want this. Okay, so we'll come back to this in a while. So what are we going to learn? So we saw yesterday an excellent demonstration session from Raja, and also an overview of what you can do, and what are the analysis methods that you can do, and what you can learn from single cell RNA analysis, and how it compares generally to bulk cell RNA analysis in the second session. So today I'm going to introduce you to another learning resources in which we have taken up a publication which focuses on understanding the heterogeneity of non-small cell lung carcinoma, and using single cell analysis or single cell data methods, single cell profiling methods. So we will explore what this specific publication actually deals with, and what we can learn from this publication, and what are the analysis that we can actually perform from data that is publicly available, partially publicly available from this. So you will learn what are the kind of analysis that you can perform using Python's environment, and what other analysis methods that you can actually perform using R's environment, are very well known and popular single cell analysis package, Suret. And there are advantages of using analysis options inside Suret's bracket, or Suret's package, or Suret's module. There are also several other advantages of using similar analysis methods outside of Suret's packages and outside of Suret's models. So we will also explore those methods today. Yeah. So let's. So what do we have in this specific lesson? So we start with, in the lesson one, it starts with introducing you towards different types of lung cancer, and that will actually tell you why performing such study is extremely important. And just like any other carcinoma or any other tumor, lung cancer also appreciably heterogeneous, right? And depending upon where it occurs, the type of lung cancer can vary, and the effect of their cancer can also vary. And there are some popular or most abundant lung cancers that we can see, and you will also see several introduction and overview about some of the risk factors, some of the recent treatments and trends. Syntax, Clement, if you are getting syntax error, then I think you are actually running the install packages in Python's environment. Please start another collab with collab.to/r. And then after that, if you copy and paste, you will not get syntax error. So yeah, so you go through this and you will get a lot of information about setting up generally the importance of studying heterogeneity in lung cancer. To understand some of the heterogeneity and some options. And what you see is you will have tumor is generally very heterogeneous tissue, right? You will get a very good overview from our resources if you go back to the course and go to the second lesson. And partially, people have tried to study this tumor heterogeneity from different methods. Like, for example, let's take. I'm looking for a picture in the associated resources. I think it's in lesson 1 only. I forgot. So that should actually help you. Let it load. Yes, okay, exactly. So this is just a cartoon of how many different types of cells that one can identify or one can actually find in a regular tumor, right? This is what we actually term it as tumor microenvironment, and many of you might have gotten some introduction or at least heard of this term, tumor microenvironment. And even before the advent or even before the extensive use of single-cell RNA-Seq analysis to study and to understand this tumor heterogeneity, people have been trying to mimic or trying to do the same with the bulk RNA-Seq technology by visually picking and collecting samples from different portions of these tumor microenvironment, right? And you can see examples of such studies all over the research and all over the databases and repositories also. That can also be done. And even in spite of those efforts, we have to remember that tumor is extremely highly heterogeneous environment, and sometimes the advantages of single cell profiling just overtakes the resolution, overtakes some of the other analysis options that bulk cell RNA gives us. So we again will have some complexities associated with this, and this publication is an excellent example of what you can achieve from such studies. So let me zoom in here. This is one of their important results that they observe. So they identify something like very close to 9 different subtypes, or 11 major cell subtypes from 42 patients that they obtained the samples from. So this is a projection, UMAP projection representation of how different cell tissue types or different tumor tissue types or tissue types from this microenvironment can be represented in the UMAP projection of their expression data. And again, this is how all the 42 patients will actually compare in the same representation. So you will get a good idea about some of the discussion that we had yesterday, right? The clustering or identification of different cell types is actually done in the original space. But since this is just a representation, you will see some overlaps which should not be mistaken into actual overlaps between the cell types or actual overlap between the data. And you can see the percentage of different tissue types populating different patients, and we can see how different these patients are depending upon their composition of these cell types. These are prominent results that this publication actually achieves, and in addition to that, they also identify some new cell types, and they also find markers, gene markers for novel cell types and cell identification using the single cell profiling method. So these are all extremely important results and very useful results that we have to discuss, and interested people can go and read this paper. This is excellent paper for this. So how to access this data? So we also have in the second lesson, we also have information on what is the data available and how to access the data, and what is that we are going to consider, and what is a project setup that we have actually set out to work with in this data. So the overall case study can be downloaded from this gene expression omnibus dataset, and and this is the accession ID. You can actually go to this by just clicking this link, which you will get this link. So here, immediately you see that in overall design, say that the raw data or the FASTQ files data of each and every one of these 42 samples is not available because of patient privacy concerns. So, so sometimes we are not, we will not get this, but in such cases, we will get access to their normalized data or processed data. So that's what we are going to get. So this study includes 42 patients, 42 samples, and you will see the same. For each one of these patients, if you click this ID, you will go to their expression table, which you can download. So this is what we will be accessing, right? So we cannot actually, or we can do it, but if we want to perform some analysis on all the 42 patients, we will need more resources, and so we restrict ourselves in this case for demonstration purposes to 5 of these samples. And these, yeah, and come down here, yes. Oh, what is this? Okay, let's go back. I think it's in the third lesson. So we restrict our study to 5 of these patients' data. So that we can swiftly perform this analysis and also study and also see if the kind of tumor heterogeneity that the original publication actually achieves in using all the 42 samples can be reflected on or can be found from the from these 5 samples alone. So we randomly selected samples of 2 carcinoma type, and one is a small squamous cell, and the other one is adenocarcinoma. 3 samples in one and 2 samples in the other, so that we can try to see if we also observed similar dispersion of different cell types, and what are the cell types that we can identify, and how different are our markers and cell type annotation to the original publication. So that kind of analysis that we can actually perform by using a subset of samples. So of course, you can select completely different subset and identify if the comparison is actually valid, and if we get similar results to the original publication, or if not. So several different parameters actually define how you get to these results, and you will see that in a while. So and you will also see learn to appreciate the complexity of single cell RNA profiling, single cell RNA analysis, unlike the bulk cell counterpart, where most of the work or most of the analysis tools are very well established, and it runs like a well-oiled machine. Like, you just start an analysis and then you will get differential gene expression analysis at the end of it, if your samples are really very well separated. So that is what we set out to